How were copy number variations discovered? Imagine looking at a giant puzzle where some pieces are missing or extra pieces are added. That's kind of what scientists saw when they first looked at DNA. They noticed that some parts of the genome didn't match the usual pattern. These differences are what we now call copy number variations. But how did scientists figure out these changes in the first place? Let's take a journey through the discovery process. Long ago, scientists used a technique called fluorescent in situ hybridization, or FISH for short. This method involves attaching tiny fluorescent tags to specific DNA sequences. When viewed under a microscope, these tags show where the DNA is located. If a piece of DNA is missing or duplicated, the fluorescent signals will be different from what's expected. This was one of the first ways researchers could see large changes in DNA copy numbers. Another early method was comparative genomic hybridization. This technique compares DNA from two different samples. It involves labeling the DNA with different colors and mixing them together. When the mixture is placed on a special slide, the colors tell scientists if a certain DNA segment is present in normal amounts or if it's missing or duplicated. This method helped reveal many copy number variations across the genome. As technology advanced, scientists gained the ability to analyze DNA in much finer detail. The development of high-throughput genomic sequencing was a game changer. Instead of just looking at big chunks of DNA, researchers could now read the entire genome base by base. This allowed them to spot even tiny copy number variations that were previously hidden. Techniques like bacterial artificial chromosome arrays and next-generation sequencing made it possible to find thousands of these variations quickly and accurately. In recent years, computers and software have become essential tools. Researchers can now analyze huge datasets to find patterns and link copy number variations to health conditions. This has helped us understand how these variations contribute to genetic diversity and influence disease risk. For example, some copy number variations are linked to congenital disabilities or certain types of cancer. Understanding how copy number variations were discovered is important for family genetic testing. It helps us see how genetic differences can affect health and disease. Identifying these variations can guide doctors and families in making informed decisions about health risks and treatments. So, from simple microscope techniques to advanced DNA sequencing, the story of discovering copy number variations shows how science keeps pushing forward to understand our genes better.